Dear colleagues, I wish to thank the organizer of the meeting for my invitation to it. I like very much Cold Spring Harbor Symposia for the very high scientific level. I participated in a number of Cold Spring Harbor Symposia quantitative biology, and probably I could also be on a couple of other such symposia, but I could not attend the meeting because of the interference of KGB. Now again, unfortunately, I cannot come to your meeting, but in this case, that's due to the courtesy of some American officials. I don't know from where, maybe from CIA or from somewhere else. Now let me switch on the presentation. Now about nuclear DNA-like RNA. The major part of this work was performed in the Institute of Molecular Biology, now Engelhardt Institute of Molecular Biology, in favor of Vladimir Engelhardt, the founder and director of the institute. The main collaborator in discovery of DNA was Veronika Mantieva. We were interested in the nature of nuclear RNA, and we isolated RNA with the aid of phenol technology. When cell suspension in saline was mixed with phenol pH 6 and shaped and centrifuged, then the intermediate layer, which was formed between water and phenol layers, unexpectedly contained cell nuclei serving their shape. These phenolic nuclei have nucleoli and chromatin, that is a photograph, and they contain DNA, histones, and other chromatin proteins, and nuclear RNA. Nuclear RNA could be isolated from these phenolic nuclei by treatment in the same way, but at 65 degrees Celsius. This RNA contain 28 and 18S peaks and also heterogeneous material. The base composition of this RNA, actually G plus C to A plus U ratio, was intermediate between DNA and ribosomal RNA. Therefore, we suggested that this RNA is a mixture of ribosomal RNA and ribosomal RNA precursors and DNA-like RNA. The first fractionation experiments performed in 1961 supported this idea. But the most clear results were obtained using the hot phenol fractionation protocol, where the phenol nuclei were treated by phenol saline at different temperatures. At 40 degree temperature, the ribosomal-like RNA was isolated, which contained ribosomal RNA precursors, and you see the base composition typical of ribosomal RNA. And in the interval of 55 and 65 degrees, the pure DNA-like RNA was obtained. The important point is that both total RNA in this fraction and nascent RNA obtained by p32 incorporation were the same, has the same base composition, actually DNA-like base composition. Ratio of DNA to RNA in phenolic nuclei was about 1 to 1 to 5. Next point is to study the properties of the nuclear DNA like RNA. Molecular mass distribution of total and nascent DNA was rather heterogeneous, and significant fraction was of high molecular weight. But an important point is that size of nascent RNA was much higher than the size of total RNA, indicating the cleavage of nascent RNA inside the cell nucleus. Also, the size of nuclear DNA was compared with the size of cytoplasmic mRNA. We elaborated in this paper the technology of visualization of nascent DNA of cytoplasm using the partial inhibition with actin-myosin D, which selectively inhibited RNA synthesis. And you can see here that nuclear DNA, nascent nuclear DNA, is much higher than, has much higher molecular mass than cytoplasmic RNA. Finally, we look for the hybridization competition properties of nuclear and cytoplasmic DNA, and we found that hybridization of cytoplasmic mRNA is completely inhibited by nuclear DNA, while the hybridization of nuclear DNA only partly inhibited by cytoplasmic messenger RNA. Therefore, we suggested that nuclear DNA is a high molecular weight precursor of messenger RNA, and during this conversion, during the maturation of messenger RNA, the significant degradation of RNA take place. Then DNA, nuclear DNA, was renamed to hnRNA by American authors. Now about hnRNP. The main collaborator in hnRNP discovery was Olga Samarina. To study the state of hnRNA in the cell nucleus, we use a milder procedure, isolation of nuclei in sucrose solution, and we extract such nuclei with such a solution at pH 7. The material obtained has a base composition intermediate between dRNA and rRNA. Then we extracted with the same solution but at pH 7, 8 to 8, and in this case we obtained a significant amount of pure dRNA, and in this case the base composition of total and of nascent RNA was exactly DNA-like. In ultracentrifuge, this RNA was concentrated in homogeneous 30S peak. The RNA in the 30S peak was of low molecular weight, and this was in contradiction with the high molecular weight of dRNA, which was shown earlier. To solve this contradiction, we use the RNase inhibitor added to all from red liver supernatant, added to all solution for isolation of RNA particles. And really, in this case, we obtain a number of peaks, the minor 30S peaks and many heavier peaks, up to the material of about 200S. Now about the properties of HNRNP particles. If to treat them by mild RNAs, they give one homogeneous surface peak. Surface particles were of about 200 angstrom in diameter. If to look of the material in the surface peak, you have just monoparticles. If you look at 45 S peak, then you have dimers. In 70S you have pentamers, and in the material of 90-100S the particles consist of 9 monomers. In all cases the bond density was 1.4 gram per ml. That is corresponds to the RNA to protein ratio of 1 to 4, 1 to 5. Interestingly, the molecular weight of RNA isolated from particles of different size correlates with the size of the particle. But in general, it was observed that the size of RNA per monomer was the same, about 700 nucleotides per 30S particle. It means that the RNP consists of a number of 30S particles linked with RNA, which is the most sensitive to RNA's treatment. Now we looked more carefully at the structure of HNRNP particles, looking for monomers. All RNA in monomer is RNase sensitive, suggesting its surface location. If to label protein with iodine, labeled iodine, and dissociate particles with 2 molar sodium chloride, then we obtained the pure protein particles with a sedimentation coefficient about 30S. So the sedimentation coefficient did not change. And these protein particles could be reconstituted after removal of 2 molar sodium chloride. You can see here original 30S particles contain RNA and protein and density 1.4, but after treatment with 2 molar sodium chloride, 30S protein particles were obtained and just pure RNA. And these 30S particles have density 1.34. All type of particles, original ones shown on previous slide, and 30S protein particles and reconstituted particles often have the same electron microscopic picture, and they also have the same properties. A number of properties were the same. There is a model that each 30S particles consist of protein globule and RNA wrapped on it. 2 molar sodium chloride removed from the surface of the particle, and then in reconstitution the original particle is appeared. The conclusion is that nuclear HNRNP is a chain of long DNA which bound to a number of similar protein particles. Each of these particles contains about 20 polypeptides, and according to other authors, these polypeptides are represented by 6 different proteins. That is about the major structure organization of 30S of HNRNP particles. Then I worked on different subjects. However, in 1990, I organized the Institute of Gene Biology, where one of the major directions is the control of gene expression on the level of hnRNA synthesis and mRNA export, or the continuation of early studies. The second main direction is the new approaches to the molecular therapy of tumors, and time in this field. But here I would like to mention briefly some results in this field, just to make some link between the past and present time. First is the role of insulators in control of gene expression. It was shown that identical insulators strongly interact and that these interactions play an important role in control of gene expression. The interaction depends on binding of proteins, in dimerization of proteins present in insulators. In particular, this is the dimerization of the MOD and the DG4. Second, insulators have a polarity, and so it means that they bind only being located in the same orientation. This determines the construction of the loop, and the construction of the loop in its turn determine the activation of certain gene. So here enhancer activates gene B, here enhancer activated gene A because of such structure of a loop. Then the super long distance interactions were discovered in Drosophila cells. They were up to dozens of megabases and even between non-homologous chromosomes. This depends on the binding of insulators. If you remove one of these insulators, this or this, then enhancer immediately stop to activate the gene. These super long interactions may influence the configuration of chromosome and control of gene expression. And finally, insulators are able to bind both promoters and enhancers. Promoters in less selective way, enhancer in more selective way. This may be the explanation why insulator located between the enhancer and promoter has an uncoupling effect. Actually, it interacts with the both and form non-productive complex. Then a number of different proteins which are responsible, which are involved in control of gene expression were discovered. The most interested are enhancer yellow 2 or any 2 and SAPE. SAPE forms a supercomplex consisting of initiation complex TF2D and remodeling complex SWICE SNF bound together with this SAPE. Removal, knockdown of SAPE led to the low recruitment of TF2D and SWICE SNF on promoter of the gene and to the inactivation of transcription of the gene, many genes. The model is that this complex binding to promoter, in which may be covered with nucleosomes, induce the movement of nucleosomes, and immediately while the promoter becomes free because of their close location, TF2D is combined with promoter. So this immediate binding of TF2D to promoter is important for the beginning of transcription. On the other hand, studies for NE2 show that it is multifunctional protein. It is a component of Saga complex, actually of its dub domain, and in this way it is involved in activation of gene expression. It is a component of THO, protein complex involved in HNRNA elongation, binding to HNRNP, and mRNA export. NE2 is also a component of Drosophila complex AMEX, binding to HNRNP and playing a crucial role in mRNA export. In fact, the knockdown in NE2 leads to the retention of mRNA inside the nucleus. You can see here. In control cells, it is exported to the cytoplasm. Here it is all present in nuclei. Finally, Ne2 is a component of several insulator complexes, fulfilling the barrier function of insulator. So that is not all, of course, results. Many other results have been obtained on the mRNA control of mRNA synthesis, and this shows the link between the seminal seminal early results and present day results. If you have some questions, they could be mailed by this address. Thank you for attention.